Friends, welcome to Biology Made Easy. Today in this video, we'll discuss some more examples of natural selection. In the previous video, we have discussed also some examples, that is industrial melanism, then pesticide-resistant mosquitoes, and sickle cell anemia. Then some other examples we'll discuss here. So the first one that we will discuss here is antibiotic resistance. Antibiotic resistance. This is very important because all of us should know it, because it is required in our life also. So you know that when a person suffers from some bacterial disease, the doctor advises to take some antibiotics. What is the purpose behind it? The antibiotics will kill the bacteria present inside the patient. But sometimes it is seen that there are some bacteria which resist to that antibiotic. That means some resistant bacterial varieties are there which can resist antibiotics. But how it happens? Whether they are mutated later on or they are present earlier, or what actually happens? Now to explain it, two scientists, Joshua Lederberg and Lister Lederberg, they conducted one replica experiment. So one is Joshua Lederberg and the other is Lester Lederberg. They conducted one experiment in 1952 which is known as replica plating experiment. So from this it will be clear actually what happens and how these antibiotic resistant bacteria actually they survive. Now usually when bacteria are grown in culture medium, what happens? Usually in one culture medium, all the medium that is required, they are taken along with the semi-solid agar medium. With agar medium, semi-solid agar medium usually taken, and the bacteria they are allowed to grow here. This is the normal practice. So what these two scientists did in their experiment? Now in one plate, they inoculated bacteria, that means inoculation of bacteria. So in this medium, this is also agar medium containing all the minimal requirements, and here they inoculated the bacteria in different points. So see, at a different points they are grown. Now this is known as master copy. This is the master copy where at 4 different places the bacteria have been inoculated. Now since the medium is provided and agar is provided, now they will grow. That means they will multiply by mitotic division. So when they will divide by mitotic division, now this will also multiply and it will grow. Then this one also will multiply, grow. That means each type will multiply and grow. Then next what happens? Now they have multiplied by means of mitotic division. Now from this copy, some replica copies are made. So how this replica copy is made? Now this is another minimal medium where the medium is there for the culture of this bacteria. But how it will be taken to that place. For this, one velvet disc is used, which is attached to a wooden. So suppose this is one wooden frame, and to this wooden frame one velvet disc is attached. This is the velvet disc. It is attached. Now this velvet disc will be simply put on this master copy. As a result, what will happen? Some of this bacteria that will be printed on this velvet copy at the same place. Just as if there will be some ink spot here, and I will put the other palm there, then automatically one print will come out. In the same way, when this velvet disc is put on this, now that will be printed in this velvet disc at the different places, just like there it will be printed. Now when this velvet disc will be put here, this is known as replica copy. This is replica copy, and you can make several replica copies also. This is one replica copy and this is one replica copy. Now you put this velvet disc here, and this is only minimal medium, nothing else is there. Now what will happen? Here you will find that the bacteria coming from this place, that is gradually growing, and this one also is growing in the replica copy. Then this one is also growing in the same place, and the other one that is also growing in the same place. This is the replica, at same place they will grow. But you should look to this replica copy. In this replica copy, in addition to the minimal medium, penicillin is also given. Penicillin, it is an antibiotic, so this penicillin is given to the medium. Now when this velvet disc is pressed against this replica copy, now all this bacteria, they will also be there. But when they will be multiply, it is seen that suppose only at one place, at this place the bacteria will grow, and at the other places the bacteria are killed. What does it mean? It means this type, the red type of bacteria that has been taken from this place, that is resistant to penicillin. That means even if penicillin is there in this medium, it is resistant to it and is capable of growing and multiplying. While the other three forms, they have been killed. That means these three bacteria, they are, I can tell that is they are susceptible. They are susceptible to penicillin, while this is resistant to penicillin. Now one question comes, where from this penicillin resistant bacteria came? Now it is seen that means this penicillin resistant bacteria was present in the original bacteria colony. It was mutated. The normal bacteria has been mutated and it has become resistant. But it was not getting a chance to multiply because other bacteria were there. But when antibiotics killed the susceptible ones, now this bacteria has selective advantage. It has selective advantage over the susceptible ones. So when the susceptible ones are killed, now this resistant type of bacteria, they multiplied themselves. So this is how the resistant bacteria to different antibiotics, they are present. I will give also another example. Sometimes doctors advise that you take this antibiotic for 7 days. But sometimes people, what they do, after 2 or 3 days they feel that I am okay now, so I will not continue the rest of the medicines. So in that case, what will happen? All the bacteria that were responsible for causing the disease, they have not been killed. Few have been killed. But when you stop the antibiotic, the bacteria which still remain in your body, they become resistant to that antibiotic. And later on, you will find, even if you use the same antibiotic, the bacteria will not be killed. This is also one of the reasons. So we should be very careful while dealing with these antibiotics. So this is one of the examples how the nature is selecting the antibiotic bacteria. Now let us give another example. Another example is glucose 6-phosphate dehydrogenase deficiency. The second example is glucose 6-phosphate dehydrogenase deficiency. This is also a very good example, and commonly it is known as G6PD, commonly glucose 6-phosphate dehydrogenase. This is an inborn error. That means some people, they suffer from this disease because of the defect lies in the gene. And this disease, it is also known as favism, because when the people suffering from this particular error, when they take beans, there is a haemolysis, and the persons will die. Similarly, if they will use anti-malarial or anti-malaria primaquine, also they will cause haemolysis and they will die. Why? What happens actually? Actually, the haemolysis is due to hydrogen peroxide. Because this hydrogen peroxide is not destroyed. That is why it causes haemolysis. So why this is not removed? Because the deficient glucose 6-phosphate dehydrogenase persons, they do not produce NADPH2, which usually removes this hydrogen peroxide. So those persons which are deficient of this particular enzyme, the hydrogen peroxide will be accumulated in them and there will be haemolysis. Now the question is, if it causes haemolysis, why such persons still survive? That is a question. Now what is the answer? Now those persons which are deficient in this glucose 6-phosphate phosphodehydrogenase, they will never suffer from malaria. Because the malaria parasite cannot complete its life cycle. That means the physiogony that cannot be completed in the persons, those who are deficient in the glucose 6-phosphate dehydrogenase. As a result, they are resistant to malaria. That means malaria will never affect them. They will never suffer from malaria. So even if this is an inborn error, but still nature has selected them. Nature has completely not eliminated such type of persons because they are resistant to malaria. So this is how nature has selected it depending upon the condition. So these are the two examples from natural selection. Now let us discuss some examples from artificial selection. Now always in the nature, variations exist. The third type we will discuss is artificial selection. So always in nature the variations exist, and the human being has exploited it for its own benefit, both in case of plants and animals. In case of animals, the high yielding domesticated cows, sheep, goats, then ducks, and different types of domesticated animals have been produced. Similarly, in case of plants also, different new varieties have been evolved from a common ancestor as a result of selection and breeding. Now I will give a very good example how from one single ancestor different types of plants have been produced as a result of selective or due to selective nature of the selective nature. So that means selective nature, how from the single ancestor different types of plants are produced. Now let us take one wild mustard. Now the wild mustard is known as Brassica oleracea. Brassica oleracea, the botanical name of wild mustard. Now from this, how different varieties are produced, you see. Now from this, if a selection of terminal buds, if only terminal buds will be selected, it gives rise to cabbage. Now if the lateral buds are selected, then we have got Brussels sprout. Now when the stem has been selected, we have got kohlrabi, and when the leaves have been selected, then kale is obtained, and when the stems and flowers selected, then the broccoli is obtained, and when the flower clusters are selected, then cauliflower is obtained. That means from the same wild mustard ancestor Brassica oleracea, all these different varieties belonging to family Brassicaceae have been obtained. It may be cabbage, may be cauliflower, broccoli, kale, kohlrabi or Brussels sprout. All these have been obtained from one plant by selection. That means by selection and breeding, the human being has got all these varieties. So this is how, because in the nature these variations exist, just by simple selection it has been done. And all this the human being has obtained in how many years? Within 50 to 100 years. So if in 50 to 100 years, human being can obtain so many varieties from a single one, then in thousands, in millions of years, why the nature cannot select such a wide variety of plants? This is quite common. So this is all about the different examples of natural selection. Thank you very much for watching the video. God bless you, and wish very bright future.